So thank you. So let's start. So here is our agenda for this morning. So we'll start with a quick introduction and news. I will do with the welcome, the news of the expert working group, and the news of the tool WheatIS data discovery. And after that, we'll have two presentations for the travel awardee. So from Lanier and Monica. So hello, Monica. After that, we'll have a small break with the group photo. And after the break, two presentations of two information systems. The presentation of Tanner for grain genes and the presentation from Paolo about the CIMIT data management system. And after that, we'll have a presentation and a discussion about the AI hackathon we plan to do this year. So we can do a very quick roundtable. We are not a lot of people in the middle, so it will be easy. So I will start. I am Michael Allo from INRAE France, and I am the Deputy Director of URGI, which is INRAE unit working on bioinformatics. And also I am the chair of the WheatIS expert working group. And I'll give the mic to Tanner. You want to introduce? Is there a microphone? Do we have one microphone or? Yes. Okay. Thank you.
Hi, my name is Tanner Shen. I'm from grain genes and USDA in California. Thank you. Hello, I'm Pierre Marot, postdoc from INRAE also, renewing the world of the wheat, and I'm here to discover what's expected to the data integration and data management in the world for wheat.
Hello, everyone. My name is Francisca Castillo. I'm from Chile, and I'm the co-founder and CEO of a company, a biotech company, named Neocrop Technologies. We are develop new varieties of wheat and other crops with precision breeding.
Hi everybody, I'm Paolo Vitale, quantitative geneticist at CIMIT and based in Mexico.
Good morning, everyone. My name is Sylvie Cloutier. I'm a retired scientist from Agriculture and Agri-Food Canada, and I'm here because I'm on the scientific board of the Wheat Initiative and the liaison for WheatIS.
Hi, I'm Monica. I am a researcher at CRAG/URTA, which is in Barcelona, Spain, and I'm also an adjunct fellow at UWA, the University of Western Australia. I work mostly with machine learning for crop trait prediction.
Yeah, good morning. My name is Thomas Lux. I'm here for the plant bio and systems biology group from Helmholtz, Munich, and here I'm standing in for Manuel Spandant.
Thank you. And Kiwan, do you want to introduce, please? You are muted.
There we go. Hello, everyone. I'm Kivan from Rosenstein Research. I'm head of bioinformatics at Rosenstein. And for 2 years, I'm founder and CEO of a company called Netminer Limited as well. And we're building a gene discovery AI system, which I can talk about later.
Thank you. So let's move on. So about the news of the expert working group. So here is a list of the members on early career researcher. So we had 2 new members last year, Paolo and Monica. So welcome again. And we have also some new renewal of some previous ECRs. About what has been done last year, a big thing was the video about the sharing experiences about AI we made at PAG and during the half-yearly meeting. We worked with Nora for the edition of this video and to push the video online after cutting all the private parts. So I reached each partner to ask if it's public, what is public for the presentation, for the discussion, and we made the cuts. And so online is all the what is publicly available. We also have a goal last year to have a better communication. So we succeed to update the WheatIS website. Even the website is quite old on the views. The content has been updated, and also we put on YouTube the AI presentation. And about social media, we close the WheatIS X account, and I started the Blue Sky one. I was not very active, so yes, it's still ongoing. I have to take more time to post things, but yes, I created the account. Also, feel free to ask questions during the presentation. I think we are a small group, so feel free to stop me, ask questions. I think yeah, it will be the best way to do. Do you have any question about communication or the videos? No?
Great job.
Other topic about mentorship. So at the beginning of this year, the WheatIS initiative presented kind of mentorship program that each expert working group has the option to implement or not. So my feeling on that is that it's a challenging process for the mentors, for the mentee, selection, agreement, reporting. So it's something not so simple. Yeah. So we need, if we want to go to this thing, we need the interest of people to be the mentors, to be mentees, plus people to take the responsibility of implementing on coordinating the program for the expert working group. Personally, it's not my number one goal. I rather focus on other activities. I want to have this hackathon AI done this year, and I prefer to focus on that. But if someone on this expert working group want to take this coordination, it's very interesting. And if there is some ECR interesting to be mentees and senior researchers interesting to be mentors, we can implement that. But the first thing before moving forward is to have someone coming back to me saying, I am interested to coordinate that. So if you in the audience or online or when the people will look at the video, come back on me if you are interested to coordinate that. It's a big work, so we can go if we have someone very motivated. Is there any question or interest in that? So not a big interest to be mentor of mentees or to be coordinator on that? We are a small, I think it's always good to have some mentorship, but yeah, the process it's not so easy to implement. The feedback from Luigi was, yeah, it's a big thing. So I think for our group, it will be better to focus on other activities. But it's my feeling. It's open to the expert working group if someone for the co-chair want to take this thing. I am open to discuss. Sylvie, you want to say something about that, or you are in line with my summary? Yeah.
Yeah, the mentorship program is something that is offered by the WheatIS initiatives as support for setting up agreements and whatnot, but the leadership of the mentorship program is left to the leaders of each of the expert working groups. So it's nothing that's mandatory. It's just something that's offered and support that's offered by.
So thank you. I understood well. Any other comments? No? Yes, Paulo?
You said supported means also that financially supported. What does that mean?
No.
Sorry, the mentorship program is not financially supported. It's supported in the sense of administrations of setting up an agreement between a mentor and a mentee and that sort of activity. This is what the board is offering. Okay, but there's the financial support that's provided to the expert working group is based on a budget that they submit annually, and it isn't linked at all to the mentorship program.
Okay, let's move forward. So now I will give you some update of the WheatIS data. Yes.
I think maybe.
Yeah.
Thank you. Yeah, maybe we should talk about the chairmanship as well, co-chairmanship.
Yeah.
Sarah, I think, moved on. So I don't know if she's still in the sciences or not. I think she's in the transition process.
Okay.
Mario, but moved.
Yeah, but it will still be working on it. But yeah, I think we will have this. I plan to have this the discussion. But yeah, I plan it at the beginning of next year to think about that, to rotate and to change also, to discuss with all the co-chair. But perhaps with some movement before 2026, because it will be better to have this discussion before the end of the year.
Okay, yeah. So I know that Sarah is replaced by, I forgot her name, but someone else.
Okay.
And that person is now representing ensemble plans in other committees.
Okay.
So maybe we can just approach Sarah and say, hey, if that person will be interested, being at least part of the WheatIS expert group.
At least, yeah.
Yeah. With Mario, so yeah, I don't know where Mario stands actually, whether he will be involved. I know that he will be involved with the strategic initiatives at CAOS.
Yeah.
And a note there will be good. I mean, there's Bradley Wolf there generating data, for example. So if you can create a WheatIS note there, I think that will be very nice.
Yeah.
But
they say that he will be working more on wheat in South Arabia.
Yeah,
but
CAOS,
but it's not a country of the WheatIS initiative, I think. So perhaps also there is another issue, but we have to talk. It's more financial issue to if you're not member of the WheatIS initiative, perhaps we cannot pay some travel. But for the science part, I think it will be nice for him.
Okay.
I understood that he will be working on wheat and on AI, so it will be perfect.
Right.
Yeah, but I think perhaps we can have a dedicated discussion before the end of the year on that.
Okay. Yeah.
I will ask a discussion between the co-chairs and then with the liaison, I think.
Yeah, so Sylvie, will you stay actually on? Since you said that you retired?
I'm retired from Agriculture Canada.
Yes.
Not for the WheatIS initiative.
The WheatIS initiative.
Oh, okay, so you will be inactive.
You never retired from the WheatIS initiative. Okay, so thanks for the discussion. So about the tool, so here are the new development and data update we have done on the WheatIS data discovery. So For the new members, the WheatIS Data Discovery is a common tool we are developing. It's kind of Google for wheat data, and we index the data from all the wheat databases around the world, and we are improving. Dave is connecting. I add Dave. Hello, Dave. Do you hear us?
Yep, I can hear you.
Oh, thank you for attending. Okay, perfect. So we just started 10 minutes ago. We had the introduction and I made some summary of what was done last year. And we had a small discussion about to have a dedicated meeting between the co-chair to talk about the future of the membership of the co-chair and so on, with following the change of Mario and Sarah. But I will plan to do that second semester to have a brief summary. So now I just started to talk about the news of the WheatIS Data Discovery tool. So all the new development I will show you has been done on FEDER. FEDER is the same tool as the WheatIS Data Discovery, but it's for all the crops, not focused on wheat. But it's the same kind of tool, and just the interface, which is different on the database index. So all the development you will see there will be done in the WheatIS, as it is the same tool. It will be easy to transfer these new features. But I say that to you because if you go to the WheatIS.org, you will not see the last development I will show you. So as you remember, yeah, you see my no. More. Yeah. So there is there. Okay, nice. There you put your keyword. Thank you.
Is this stuff?
Sorry.
That's okay. I'm grabbing stuff too.
Okay, I have to find a good one.
Laser is not working well.
Okay, so perhaps on some screen it doesn't working because the screen.
Oh my God.
Yeah.
First there, but not.
Yes, it's because it is a screen. So no worry.
Okay.
So you put your keyword in the search, and it will find this keyword in all the metadata in all the databases around all the index databases. So it's a classical feature of the WheatIS Data Discovery, and we add additional things. So for example, we developed this map, the map you see on the model screen with all the germplasm. Where are the germplasm hosted in the gene banks? We also, and you have this summary for each germplasm with the map, the description, the breeders, the donors, some evaluation data collection panel, the link to this section in the different panels collection, and so on. And we also developed some summary table with a list of all the accession. And if you click on the export button, you could download this list in the Excel format following the standard. So for example, the MyAPI standard, which is the standard for phenotyping data. So we developed some download capabilities to help you with the results. Also, we add the possibility to order some accession. So if you click on the little basket, for example, on this Apache accession, it will open an order summary, and you have multiple accession, and then you can copy the order with the detail of your personal information, and so on and so on.
So,
Yeah.
so where do you order it from?
So it link directly to the gene banks.
Okay, so we
For the gene bank who accept to receive orders from So
Right, so
it's not for all the accession.
It's just for us, so it's European.
I think it's European. But if some US gene banks want to do that, it's possible. We have the feature available.
Do you actually do you have the Genesis?
No.
Yeah, I think if you have that link, then I think they have
also the US also
green, for example, US greens.
Perhaps to dig on that.
I don't know what symmetry is. It's used from symmetry. So how can we do this through symmetry?
Well, I mean, we have, I will present then our.
Perhaps with a microphone for Q&A, Dave, if possible. Thank you.
I'll present later our data management system, but not this specific task on writing memo lines and having a result. I think it's not implemented.
Okay, but you guys have a node already, right? So if you provide that maybe in a row where that link may need to go, then I think that may be a part of this improvement.
I think the first step is to, if it's easy from the accession card in the CIMIT database to order it, you just have to click, and you will go to the CIMIT databases and order. I think it's an easy the first step, and the second step is to add this directly in the WheatIS data discovery with the basket thing and the order things. I think it's
even if you can pre-populate the page, order the page. Yeah, if you can at least pre-populate their pages for the ordering, I think that would be good. Because there's a problem. For example, one problem with ordering seeds is that you have to log in, you have to enter all this information, right? So, but if you can just say, okay, I want this, and then at least a page populated with this ID number associated with that seed,
it will be useful for the user, but there is some issue, RGPD issue, security issue, to have some transfer of IDs. Informatics.
maybe with some links, like URL based, maybe that.
But we cannot do some cookies. I think it will be perhaps some issue, legal issue. So we have to, because this is secure thing. And we have to think, but to be sure that it's clean. And online Q&A, Dave, feel free to ask a question and to raise your hand or to directly open your microphone. We are like 10 people in the room, so it's you can interrupt and discuss.
No problem, thanks.
Perfect. So we also developed some summary of the phenotyping data set. Here's an example with trial from INRAE. And we also developed some download capabilities. You can click there to download your phenotyping results, selecting your traits, your years of experiment, and so on, to have a table, Excel table with the results. And of course, we have the link to the accession, the link to the ontology, the link to the studies, and so on and so on. So I just put a link here from an ontology variable. You will have all the detail of this variable with the name, in which ontology, the trait name, the scale, the method, and so on. And in test, we are trying to display some fair digital objects. So using the INRAE-URGI technology to index the EVA databases at TBI, and we are succeeding doing this. So it will allow to use this method to index new databases. So I don't know if you're aware of this kind of tools. No, INRAE-URGI, and so perhaps I will present that the next time it will be in production. And yeah, it's a method to describe the data with metadata fair. Perhaps Q1, you know that.
Do you have any visualization associated with this?
No, it's
just data sets, it seems like, right?
yeah,
There's no
just
visualization.
metadata and yeah.
Yeah, we do have
In
a
a fair
tool
manner.
to actually visualize diverse data, but against a reference genome. So I don't know if this wild level wheat, I'm guessing it's oh, okay, so that may work here. There's a reference genome here, right? Okay.
Yeah, it's all the data. We took all the data from EVA, the wheat data, and then we use this kind of FDO, that is this format, to describe that. It's that EVA who provided the INRAE-URGI format, and we integrate this INRAE format as a metadata index. So it's kind of like using the reading API for the phenotyping part, but it's for genotyping. Q1, do you want to say something?
Nothing too specific. I'm familiar with research object crates, and is it not a bit similar to frictionless data? It's just a way of describing data and its metadata. So yeah, it's good to see. Is that something that is already supported or is that in development?
It's already supporting, so we have done that for EVA, but Sarah is not there. But the discussion is ENA also implementing INRAE-URGI? It's not completely clear if EBI will using this INRAE-URGI for all their databases or EVA ENA.
Right.
Not for ENA. It's not completely clear because it's different teams. So yeah.
Gotcha. Yeah, I don't know.
So it's a question for Sarah, but she's not there at the moment. So the ongoing developments, it's since I think 5 or 6 years we want to do that. Nathaniel, do you remember in Paris we already talked about that, to have a mean to monitor the outgoing the traffic from the WheatIS towards the partner database. So we have the tool to do that now. We have a small database with a result, and we have to exploit this database. So it's ongoing. It will be done 5 years after, but it will be done, and I think it will be a huge improvement to give some metrics and say that you have this number of people coming to your database from the WheatIS. So yeah, it will be done in the coming months. And what we want to do is to display the date of last data update, because it's very important for the users to see if the data are up to date or not. And also we want to develop on front of the keyword query chatbot to allow some semantic search, something more easy for the users. Nowadays they like to interact with the chatbots, and so we want to have this to have a chatbot in front of this WheatIS. But it will we have to work on that. Is there any comment or question on the ongoing developments or something big is missing?
I've got one comment about
Yeah.
the chatbot. So the idea is good. It would expect or assume users to know about WheatIS and Discovery Platform to come there first and then use it. But I think where the field is slowly moving towards is that people just doing those searches more naturally in ChatGPT, in Gemini, in Claude. And I think what I will be talking about later on in the AI hackathon topic is how can we connect WheatIS as a skill into all those different new search tools that people probably more naturally going to be using, and it can still come out and hit some of the endpoints that WheatIS can provide. Say, if you know what I mean.
Okay, so what you will discuss is not to have another chatbot in front, but using the existing one with providing the data to the existing one. I understood well?
Yeah, providing connectors that it can
Connectors.
connect to WheatIS resources databases within
Okay.
within existing chat platforms.
Okay. Could be interesting. Yes.
Yeah, I think K1 also will be touching that subject, but so who will pay for that? That's my question, right? Even the processing of the prompts into large language models.
Yeah.
That
it's not
there's
free.
some money.
No, nowadays it's not free. Yeah.
Right.
Yeah,
So I don't know if Dave or K1 found a solution to that.
K1 for Netminer, he has a premium
That's
for more
maybe
token.
that.
Yeah.
For you guys.
Yeah. At the moment, the tool is completely open data and free of use, and I think it would be nice to have this. Yeah. So if we have to pay for tokens, it's not so easy.
Right.
What I and after I give you the mic, Monica. It's something perhaps more simpler. Is that for example to have some semantic in embeddings. So it is not using LLM per se, but more embeddings, and we can do that without a token, without paying OpenAI or other company. But it's more basic semantic.
Well, but you still need the tokens to process if someone asks there's a question, right?
If we are our own embedding, no, you can just host it. This. Yeah.
Okay, we'll talk about
it. Yeah, Monica.
Yeah, I just wanted to add to that because it's not free, but that is an open source model, which I'm going to talk a little bit in my talk called Llama, and you can download and run in your own servers. So you could have the chatbot possibility without having to send anything to OpenAI, and I think I like that also because it's more secure if you want to use that with data that's private. But it's not free because you still have to run your server, and the server needs to have access to GPU. But it can run in kind of a gamer computer as well for one person, not for the whole community. But I just want to add that that's also this possibility.
So just one GPU, you guys have that?
Yeah, it is. So the model itself already works really well, so you don't have to fine tune it that much. So for example, for one person to go back and forth, one GPU is more than enough. Yeah.
Okay, thank you. So we'll have to explore the different things before moving forward. Thank you. About the data, we have some updating in progress, almost finished. We have a first one of removal of deprecating indexes. And we updated the last version of ensemble plants and Grammy. The next updates plan is work on ENA. Can we use AeroCrate on that? And also I will be interested to index the AlphaFold DB the database at INRAE-URGI because there is a lot of wheat protein in this database. I think it would be nice to have an update on grain genes if Tanner want to do that, and not Netminer also. And I identified also new databases. So at University of Western Australia, there is a wheat panache. So we have to work with Dave to index this one, I think. And at IPK, there is a pan-barlex. So it's barley, but yeah, I think there is some interest to index some barley data in WheatIS. And a lot more, but I think it will be the main the focus. Do you have some comments or recommendation or on the list? Yes.
Well, I think also CIMMYT Dataverse could be added in list. I mean,
we already had the CIMMYT Dataverse, but perhaps an update should be nice. You identified some updates to do. Okay, nice. I will add it to the list. Thank you. We need people to ask. So thank you. Other comments? No? So okay.
Is this your last slide? Or?
Yeah, sort of. No, I think I have one more. No, I have some metrics. So it's slowly growing, the usage. The first user came from China, India, and USA, so it's very worldwide usage of this tool. But this matrix does not include the wheat research from the FEDER. So FEDER is the same tool for all the plants, which is mostly used in Europe. So I think there is a lot of metrics missing in there. So I will have to work to have some common metrics between the tools to have a clear view of what is used for wheat research. Yes.
Maybe you should sit here. So how did you? One of our problems to get metrics is the bots. There are so many bots coming from different places.
Yeah, we try to stop the bots. Yeah, it's excluding the bots, and for the ones who we cannot exclude automatically, I do this by hand.
Yes.
When I see 1 month with 10 times, I remove it in the but it's complicated. I use a tool called AW Stats. So it's not Google Analytics, it's another tool. So it's try to exclude bots, but it's not so easy. Yeah. But in the future, I think there will be more AIs than humans trying to harvest all the data, but with AI, they will do something. So yeah. And plans for next year, just one slide that is a kind of introduction, and Q1 we'll talk about that at the end of the morning. So we want to do this AI hackathon. We plan to host it at Rosam State Research, thanks to Q1. So Q1 will present some potential developments and to discuss what we want to do together. We have a budget secure for that for this year, but we still have to tackle some issue in the fundings. So we can fund the meeting organization. We already work with Q1 to have some estimation of the cost per person and so on. And we want to offer some travel awards. At the moment, we can offer travel awards only for ECR members. And I think, but perhaps I'm wrong, some of you who are not ECR want to attend. So for these people, we have to find a medium to give them the travel award. And it's a discussion with the WheatIS initiative, but also with JKE. So it's not so easy. It's not only science, it's also administration and so on and so on. So yes, I think also, and also there is a maximum of 1K per person for the travel grants. So if you're coming from Australia, for example, perhaps you will have to pay the additional costs. And also there is potential issue of people who are member of country, not member of the WheatIS initiative. So there is some funding issue we have to tackle. I think probably we will, some of them will tackle and other not. I don't know, Sylvie, if you have some things about that. What is the procedure if we want to have some non-ECR with travel grants? There is also some other type of awards or for the co-chair also. Yesterday I heard that the chair and the co-chairs should have the travel award also.
So there's two things.
Yeah.
One is an award.
Yeah.
So an award is awarded, and therefore that's a competition. So you
Yeah.
have to set up a competition. So you have to use grant, as you're saying there, which is different from award.
Award, okay, grant.
So travel grant, the maximum of 999 euros, that's set up by JKI.
Yeah.
Because if it is over 1,000, then they have to request further approval.
Yeah.
If it is under 1,000 euro, then they do not require approval, and that's why they said that, because that's just a blanket that they can provide the expert working group. With regard to who can be funded through award, it's definitely just the ECR. Through a grant, it could also be the chairs and co-chairs, but please verify that with Teresa to make sure that I'm not misinterpreting. For any amount above 1,000, it requires board approval, not scientific board approval, but board approval. So you can make special requests, but it has to go to board approval before you can spend anything above the 1,000 or 999 euro.
Thank you. But yeah, I think it will be important to have the names of the people interesting, and if it's all the people are eligible, it will be easy. If not, we have to start the discussion now. To try to fund to have the grants for the people. Yes, Monica. Hello, Mohamed is trying to connect. I think it's okay. Hello, Mohamed. Thanks for joining on Sunday. Yes.
Yeah. No, I just had a question about the hackathon. Do you guys have is that a question already, or like the dates and everything, and what happened?
Yeah. What is important is to have this on this year or at the early beginning of next year, but with all the expense already done or planned. So yeah, and before I think the limit is like June of next year, because there will be a change also in the hosting of administration of WheatIS initiative. So there is a hard limit. I think to be realistic, if we move forward now, we can target the end of this year, because to have time to organize the people to take their planes and so on and so on. But I think we have to move forward now to ask also the authorization and so on and so on. And the talk of Q1 will more be on the scientific part, what we want to do, and when we decide what to do, I will send a message to the group to have some kind of interest, who is interested, and to move forward with Q1 to find the date when the room will be available and so on and so on and so on.
Can I just ask, do you know if there's any other potential funding that might be available? Certainly that amount wouldn't even cover the flights coming from Australia. So if you're looking at accommodation or anything else, it's going to start getting expensive for us. Any idea on what are the costs may be involved and if they could be covered by other funding, perhaps?
For the cost? The idea is to have all the expense, the meeting organization, the room, equipment, meal, paid by WheatIS. And then there is this travel grant, but it's 1K. So if you need more than 1K, you will have to fund by, if you have some projects, to co-fund your travel, or we have to ask some special authorization to the WheatIS initiative. So in advance, I'm not sure that it will be accepted, but as you are, for you, if it's you, Dave, it will be easier because you are a co-chair. If it's someone else for your team, it would be.
Yeah, I was thinking of saying something from my team, but so you're suggesting that accommodation and possibly some meals may be included from a separate budget? Or would
Okay.
that come from the travel budget?
Oh, I don't know if we can take the accommodation, the hotels and so on. That's you have in mind?
Yeah, I'm trying to think of the overall cost to see whether it would be feasible to send someone.
Okay, but I think perhaps we have to have a dedicated discussion on that.
No.
The door is closed. Yeah.
I think he's saying that not to pass because we have this 999 limit.
Yeah.
So he's looking for ways not to exceed that by, just looking at other budgets. Maybe we can just pay like meals and other stuff or another budget item.
Yeah, but once we had all the eligibility and so on, you can look at your co-funding.
That's true.
Yeah.
Mm-hmm.
But the idea is to, yes, if we can use this money because we have the authorization, it's like 700K for that. So it can cover a big part of the event. If we are 10 or 8, we can cover a big thing. I put the majority of the budget in that.
Yeah, I just have one more question, not budget related. I don't know if they're going to be presenting more about the hackathon later, but I was wondering if it's what size are you thinking? Because I was looking at, there is the Syngenta hackathon, which was quite big for a few years, the Maze 2 Fields, and I think to attract good, people to actually come and try and do a serious effort. They had let's say a descriptive paper after describing all the solutions to the hackathon, and I think that brings more interest to people who, of course, students are great, they should do it, but also some people have a bit more experience to help them and maybe go with different groups and I don't know, have more people participating in general. I think the Syngenta hackathon didn't have papers involved, but people would present their solutions at a conference that was in a similar timeline. And the conference was always it was not a conference for them, but it was kind of tangentially related, so they could present. And then one of the prizes was the money prize was not that big, but they funded the person to go there and present. I think that also maybe attracts more people. But yeah, sorry, just had comments about hackathons because I love them. Fun.
Yeah, I think I rather focus on what we want to do. It will be a small one for WheatIS members only. Perhaps we could add more WheatIS member if someone from your teams want to attend. We can ask them to be new ECR, and we will welcome them. But I think we have to focus on what we want to do. But after that, yes, it's a good idea to have a paper or to have some advertisement for for the people to show what they have done. I think it's a good idea. We have to think about that. Yeah.
Well, that's a good point, but right? Yeah, that's a good point because you know some hackathons are just hit and miss, right? Some just like hackathon, just there's no incentive. But think about incentives are good. But and it depends on number of people who will apply for this. So if there are not enough people, perhaps we should expand it at least to other expert work groups.
Yes, it will be which initiative open, but
Okay.
yeah, of course. But for people interested in the field of what we want to do. So perhaps there is other people in the other experts working on AI, but more applied on some, for example, phenotyping robotics, and not, for example, exactly the same subject as we will do, but we will be open, of course.
Yeah. At least if they can advertise it in their institutions, maybe we can actually get those people who are more interested in the computational work, which may actually help us
to grow the group. Yeah,
and they can join our working group as what we call early career scientists.
Yeah.
I think on a practical basis, you're better off getting the people who are doing the hands-on stuff rather than the group leaders. For a hackathon, if you want a planning session, then you might want the group leaders. But an actual hackathon where people are doing more hands-on stuff, you probably get better off getting the people in the group who are doing the hands-on stuff.
Yes, it will be the idea. Yeah, thanks, Dave. Okay, so we will have time to discuss the subject with the Q&A at the end of the morning, and I think it's done for me. So the next speaker will be the travel awardees. It's you, Lamia. She's coming. Yeah, okay. Can you go there and you have an USB key or with your foundation? Perfect. Okay.
So if I send my presentation to you, that it will be here.
Yes, we have to present all from here to have the online people. Yes, you can send to me by mail or what you prefer. So we will, for the people online, we will connect the USB key. Thank you. So you will be able to take a time also to present by yourself. Okay. If you want to, yeah. Not so fast. Yes. Okay. Perfect. I can shorten that. So it's okay for you? Yeah.
Yes, thank you so much. Hello everyone. So my name is Lamia. I'm a PhD student at ICARDA in Morocco, Rabat. And I'm working on plant growth promoter rhizobacteria mediation for improving drought tolerance in spring bred wheat, Triticum aestivum. So today I'm going to work on to talk about a part of my work as a PhD, as my PhD. So, and at first I want to thank you for this opportunity to present my work. I want to start by a small introduction and give you some numbers about Africa and wheat. So Africa grows 10 million hectares of wheat per year, but producing 25 million tons as a with the drought we have, but we are consuming 75 million tons. So we are importing 55 million tons at the price of 20 billion USD. So here comes the need to develop a high yielding wheat varieties with a tolerance or resistant to major biotic and abiotic stresses. So so the first part of the work was a panel of 20-something different bred wheat lines from ICARDA's germplasm that was phenotyped and sequenced. And the two objectives of this work were the first one to uncover the significant MTAs linked to the yield-related traits. And the second one was to choose the top performing genotype, one a resistant and another one susceptible, to be able to prime these with the rhizobia I am using and see the effect of these bacteria. So at first, just some photos to show you the one in the left is a plot with a susceptible genotype, and the other one is the tolerant one. The other photo is for a drought-tolerant wheat genotypes in Morocco, and these are rain-fed. It's not with irrigation. So the results of the GWAS that we've done, we got these Manhattan plots of the 246 bred wheat genotypes for grain yield at several stations we had. And we can say that this study identified a total of 65 significant marker traits associations across the studied locations, and the highest number of pairwise markers on the B and A genomes, respectively, and the lowest number was on genome D. And this graph, it's just the marker density on each bred wheat chromosome. And we discovered 14 significant markers were identified as associated with grain yields. And this is an example for a marker that was located on chromosome 5B, recorded the highest value. And this marker was associated with 1,000 kernel weight at Mercer Station. And another study was found that this marker influenced leaf morphology, plant architecture, and yield also. And this work was already published on plant gene. So as I said at first, this work was also to help us choose one of the top performing genotypes that will exhibit both high yield and drought tolerance, and another one that is susceptible to drought. So so I can be able to prime these genotypes with the rhizobia. So here comes the second part of the study, where I will take these genotypes I chose and prime them with the rhizobacteria and see the effect of these microbes. But I want to say that over a century, researchers have investigated the use of plant growth promoting rhizobacteria as an alternate technique to improve drought tolerance. But despite all this work, the interaction of rhizobia with non-legumes was generally overlooked as a research topic until the last decades. So in the present study, we aim to improve wheat growth under drought stress through priming these genotypes with the rhizobium. So directly to the methodology, it was the preparation of the bacterial suspension, the sterilization of the seeds, then it's the priming, all the tests that follow, the planting also, and at a certain stage, I introduced the drought. So, so for the drought, I had 4 blocks, if we can say. The first one was a 100% field capacity, a 75% field capacity, and the drought part was a 50 and a 25% field capacity. So for the data, we collected a set of morphological and yield-related traits, biomass traits, yield, tillering traits, seed morphology, and also physiological and biochemical traits. So for the results, we can see in the blue part that irrigation is have a significant effect on most of the traits, but what is essential for us is the effect of strain. So we can see that the strains have a significant effect on number of seeds, which is the NS, 1000 kernel weight, and the tillering also. So here to just to show you the effect of the strains used. So the yellow part is the control, the rest is the strains used. Here is just the irrigation regime, so the 100, the 50, 75, 50, and 25. And the first graph is for the number of seeds, and the second one is the number of tillers. So we can see, for example, in this first part that bacteria number 1 had the highest, if we can say, value of an effect on the number of seeds compared to the control. And of course, the rest of the strain follows. For the number of tillers also, we can see that there is a positive effect of the strains in these 2 blocks, but not really in these 2. So it's not really working under port droughts. So we did also a field trial. It's also strain preparation, seed priming, planting, but this part is a bit different than the pot experiment because it's the field, so it's different protocols. And oh yeah, and in this part I need to do another if we can say validation, a second year data, but this year it was not dry enough to get any, fortunately. I did also a microbial whole genome sequencing with the Get Genome project. I am analyzing the data right now, and I think it helps to understand more the mechanism, because even if, as we said, the rhizobium is usually used with the legumes, but not the non-legumes. The legumes, we know the mechanism from A to Z, but with the non-legumes, it's still not clear. So maybe with the whole genome sequencing, we can get to understand the mechanism with the annotation, etc. So we can get more details about the potential. So the perspective, as I said, the field validation to get the second year data, the strain characterization, and the RNA-seq analysis of wheat genotypes, the inoculated and the non-inoculated. So due to the wheat genome is really, really big, so it's a bit expensive to do so. I'm keeping the samples, and maybe I can do that maybe later when I can find it. So to conclude, overall, these approaches will help us better link microbial traits with plant responses and move toward more efficient and sustainable agriculture strategies. And also, we are not aiming to replace fertilizers with these biomethods, but at least to reduce the usage of the fertilizers. And I love to finish with this photo, is that they live longer than us, so maybe we have to try to keep it fertile and clean for them. And thank you for your attention.
Thank you for the presentation. Questions in the audience?
Thank you. Thanks for signing up for the I think it's an attendance sheet, but it will work as an attendance sheet, and also you'll give me good permission to be in the picture when we are taking the picture. Question. So your first slide was interesting to me, that there is a like $50 billion gap. Is that it?
Yeah.
Between the produce and use. So what's it? So yeah, we are scientists essentially, but so what is do you think that the origin of this? Is it because people just not adapting to this, the right wheat? Or is that there's no right wheat available? I'm sure there is. Or just like the knowledge gap. And there are some extension programs, for example, in Europe, in the US, other places. So what do you think the cause of this?
So first of all, is the climate change. So it's drought. So you can't get what we need because of the climate change. And at the same time, sometimes even if the breeders generate new varieties that will give more yield, sometimes the farmer don't want to change the variety because he is using it for decades, and he knows that variety is working. He is not ready to change the variety just because scientists said so. No, he needs to see the results. So now with ICARDA field days, and they are trying a bit to, they are trying to bring the farmers to the field days so they can see by their own eyes that these varieties that we are generating and these products that we are generating are giving more yields than the variety that you are used to.
I don't know if you're familiar with any of the country by country basis, like, because you talk about adoption, right, of new species. Are like some countries better in adapting, Morocco or Tunisia, than others, perhaps?
Yeah, I think so also.
Thanks.
No worries.
Thanks for the presentation. I have a question. Probably, I didn't pay attention because I just received the paper to see in the full moment. But I saw that you have done an experiment in, outside the field and in the field, right? And the one in the field, did you also collect in the data, and there was some impact of the strains on yield? I didn't check it.
Because I didn't talk about the data, you didn't miss anything. Actually, we did have great results, but to validate that we need a second year data. And this year, I did the experiment, but it was not dry enough to see the effect. It was there were a lot of precipitation, so we can see the effect. So the irrigation, if you can say, cover the effect of the strains. So we didn't get any good results, if you can say.
Yeah, thank you for talking. I just have one question. I don't know if you've covered that. For example, you did the priming and then tested the seed in the field to grow. Did you check, or are you guys going to check if there's any change in the microbial community in those seeds that were primed, like after the plant's growing, to see, just to see if the colonization is successful, or if there's any influence in the microbial community?
Actually, after the inoculation, we did just the test of the viability, but to check the the community, we call that, bacterials in the soil after the inoculation. We didn't do that. But I think it takes a lot of time to do so, but we didn't cover that part. We just tested the viability of the inoculum after several days.
Makes sense. First, we need to know if it's
Yeah.
changing. I was just curious because I imagine that introducing this microbiome is not there in the soil like usually, or at least in that concentration, how that would affect. Yeah.
I see, but the issue in this area is the viability of those, because it's the problem or the issue is to keep those bacteria alive, because in several experiments, they when you check the viability, they are not there anymore. No worries.
Thank you very much. Thank you for coming.
Thank you for the opportunity, and I'm sorry I'm late.
No, I think Monitans, you have a USB key, or you send your petition by mail? What?
Yeah, can I put up the line?
It will be complicated.
Already.
Okay. Yes. Do you have an USB key, or? Let me can, because I'm plugged.
Yes. Yeah, thank you.
Perfect. Okay, so I'm an adjunct fellow at the University of Western Australia, and I work at INRAE-URGI, which is as a researcher, which is in Barcelona, Spain. So I'm now most basically in Spain, but also working with UWA, and that's where I also work with wheat research. So my main line of research is actually working with machine learning and developing machine learning broad models for breeding, and to try to build better plant representations. And that involves different types of data sets, and try to collate these data sets in a way that we can represent a little bit better what the plant is going through and what genetic resources it might have, and the G by E interaction and things like that. So that involves using all the data usually that I have available on those trials. So to start off, I'm just going to present one of the works that I did with multimodal deep learning, actually that I had one of my students doing, a master student, Rohini. She did a great job in that. And the goal here was actually to collate the data that we had from a paper. That's actually the data was collected at CIMMYT in Mexico. And it was data from 2 field trials of wheat that they collected the multispectral images from those plants growing, which was input in one part of the model, and also the genomic variation data, which was in the second part. And to try to bring those things together, which I think one of the parts that I'm more interested in, I built a machine learning, a multimodal model. So the first part of the model, the green part, is actually processing the image data. So it gets all those multispectral images collected by drones together with some vegetation indexes, which is basically just use a few equations to combine those bands and trying to get better, a better image of specific traits. So for example, you can see if there's variation in leaf color that indicates stress, if there's variation in leaf color that indicate maybe a higher accumulation of chlorophyll, and different things like that. So when you kind of calculate the difference between some of those bands using the vegetation index equations, you're able to better represent some specific patterns of the plant. And then we use that to train the image portion of the model. And the blue part is actually showing the portion of the model that was dedicated to the genotypic variance, which in this case was SNPs. That was all we have. The SNPs were already filtered for linkage disequilibrium and some correlations. So this is mostly representative SNPs from that population in that data set. And both of those models, they extract the features separately. So they try to understand what those features mean for the crop yield.
And then after that, we had the diffusion layers, which are those three at the end, in which both of the features extracted from the different sites are merged together, and the model tries to see the correlation between them. So we built this model for grain yield prediction and testing. We tested, like, we compared the genomic part with the Bayesian regression model, which performed quite well, which is the BGLR, which performed just with genotypic data, was able to identify the yield, like predict the yield with 75 correlation. But the unimodal deep learning models, for example, when I got the metrics from just each one of those portions, it didn't perform that well. The genomic model was 0.4, the image only model was 0.65. But when I put them together, you can actually see, I think, the better power of the machine learning model, which is not only trying to extract the correlations, which, for example, we can do here with some linear regressions, but how do they combine? And what does it mean, for example, if the plant have a canopy of this density with this kind of reflectance values associated with the genetic background? So this is what the multimodal model kind of puts together in its clunky way, but it tries to put those features together. And then the prediction was quite good, was 0.84. And the model was able to place those individuals from the trial, which were about 8,000 individuals. I have to just thank CIMMYT for having such big trials that have a lot of data, and it's really good to train machine learning models because that's what we need. And the model was able to place those individuals into categories, so which individuals were more likely to have a low yield, medium yield, and high yield. And the idea here is that maybe you can select for the high yield to continue the trial or to plan your crosses. And I mean, you can do that for different traits. But the model not only predicts, I think, the actual yield value, which is interesting, but maybe not that useful because always going to be a little bit off. But it can classify also to different buckets, so you can know what you're going to do with each one of them. And after that, we used SHAP, which is an explainable AI model, to try to extract which SNPs were more interesting for trying to find associations with yield prediction. So it's quite similar to what a GWAS would do, not the plot, but the idea is basically the same. So in that case, for the model and for its prediction, having those SNPs, for example, this is the SHAP value impact, and this is the feature value. So for feature value, 0 was homozygous to the reference. The middle here, which is 0.5, was heterozygous, and high was homozygous, but different from the reference. So what it's saying here is that when I have a homozygous different from the reference, this is not good for yield prediction. Oh, yeah, this is not good for the yield prediction. So it has this value on the negative side of the SHAP, which is marked by this bar. So anything on the negative side, it means it influences the yield prediction negatively, and here it influences the yield prediction positively. So I can see here, if I have homozygous to the reference, it seems good for a higher yield prediction. And then you can look at those, like all of those SNPs. So you can see we have quite a few in 7D, different spots, but kind of close by, in chromosome 4E, 4A, more in 7D. So it seems like to point out some of the regions that seem to be more associated with a higher yield or lower yield production. And one of the questions that we have usually when we do this machine learning models and try to get some biological insights from the predictions is if that makes sense, actually, or if it's just a bias within the model, within the population. But it doesn't mean actually that those have any association with the yield.
I have a question.
Yeah.
So I'm not very familiar with the SHAP value. Does this work now? Hello?
Yeah.
So yeah, I'm not familiar with the SHAP, but so if you just go to look at the first and second row, does it mean that the absence of the second SNP is actually increases or better predictor?
Yeah. So for example, in the first row, we have mostly homozygous. So yeah, like I said, like if you have a homozygous different to the reference, you have a lower yield, homozygous similar to the reference, higher yield. And yeah, the second one, it kind of shows maybe a dosage effect of that. So if you have a heterozygous, it stands kind of in the middle. But if you have homozygous different from the reference, higher yield. So it would be like maybe one allele already provides some contribution. And for example, here in the, I think that's the fifth row, you can see that having heterozygous provides a lot of contribution. Like most of the heterozygous seem to be here in the middle. And then you have this effect a little bit. And this effect also like the length of the effect is because they might have different contribution, like we have a contribution value per individual. So each dot would be one individual, and when they cluster kind of together, it means more individuals seem to have the same relationship, same feature relationship for their prediction. So we have some dots that are extended, which means that for some individuals, for example, this SNP here on 2A seems to be quite important for yield prediction, but not for all individuals. So SHAP is really good in showing you kind of the directional variation. And also then you can see for how much of your population. And because these results can be disaggregated, you can look at specific, for example, if you know the pedigree of them, if you know their the population structure, you can look within different populations, what's the direction of the SNP contribution. But yeah, we don't know how biological that is. Hope that is. We're looking to see actually if we have any genes close by and things like that, or if people have found any evidence for those SNPs. And that brings actually to the second part, which like we're integrating different types of data to try to represent the plant conditions with as many layers as possible, as much as we have available. But that also means we still only work with data that we have at the moment. And that's quite restricted because of well, how much it costs to actually generate this data. It's expensive to get someone to fly the drone. The cameras are expensive, at least for the first buy, not to continue use. But genomic data is expensive, expression, everything. So we had this project actually with Dave, and this is another master student that actually developed the prototype, which was just using the language models to try to enrich those data sets that we had and also validate what we are finding as the prediction. So what are those SNPs related to? So the first idea was just generating this experimental pipeline for the data extraction. So turning the unstructured data, the thousands and thousands of publications, into a table, a structured table with a controlled vocabulary, and using that for then generating knowledge graphs, or you can use for graph machine learning, and to then try to enrich the data set as we had with, for example, the genomic data set, genomic variation data set that we had. So like I said, this was made by a student, Stefan, and we used actually the disease resistance as our question. So we were looking at different disease resistance genes in legumes, because that provides a much also smaller data set than if we try to do the same thing in wheat. That would be way too many papers to clean and filter and do everything. So we're looking in legumes, and we were able to find like from 1,500 papers that were from the Scopus Query. They were downloaded, parsed, transformed into raw text. The raw text was automatically screened for relevancy. So we have 779 papers at the end. Those were split into test, train, and validation data sets for testing the language model. And like I said, we're using this for disease resistance, for finding disease resistance genes in legumes. So we had a few questions that were developed by the expert group that works with this, which is Jacqueline Baffley group. That were actually what were the questions that they wanted to extract from this data set. And like I mentioned before, so we use Llama, which is the open source model that I mentioned, because when dealing with this kind, like with the papers, we were, there is a constraint regarding copyright. Like we as an institution have access, but for example, when we publish this, doesn't mean like people will have, like I will have access and want to be responsible for how do they acquire and use their papers. So we're using Llama, which is an open source closed model. So you can download that, you can run on your own server, and you are kind of responsible for the questions and queries and how much tokens you want to use. Because it's free for running for yourself, but of course, the bigger your questions or the more complex they are, the longer Gamma is going to take to run. And we don't want to have to provide server for running very complex questions that take a lot of time. So this is kind of the extraction pipeline that we're doing. So all of this for this prototype, it's all automated because even when we're dealing with just 1,500 papers at the beginning, it is quite time consuming to read and separate them and see what they're associated with. So all of this was done automatically using like APIs, different custom Python pipelines, and different prompts. So we did use, of course, we did use Gemini for building our gold standard data sets. So we used Gemini to do automatic extraction of the things that we wanted. And then we manually checked those, which made everything much faster. And then we compared how Llama performed based on that golden data set. And this is, for example, an example of the fields that we were trying to extract. So we had the record ID. This was actually a metadata that was imputed into the chunk, which said which paper it was. We had some of the keywords from that paper and anything that could help the model kind of identify and help us identify where it comes from. So all of the responses would be tagged to the paper, which part of the paper as well, which paragraph. So people can, once they have the full structured data set, they want to check something or they want to learn more, they can just go and find exactly where this information was and which paper it was. So it's easy to validate and also easy to continue exploring. The other information that we wanted was the gene name, aliases, species, cultivars, all of this information below. And this is an example of how the model performs. So this is just a small example for it. So we had a little bit of problem actually trying to establish the metrics for this model because semantic quantification it's quite hard. So the model would, for example, in this first one, I think that is a good example. So for the model, it outputted the confidence level of the gene, not of the model. So this was a putative gene. It was in this cultivar, and this is the extracted gold data set. So that's our ground truth. And this was the model extractions. And we could see that the model extracted, for example, the cross. So we found that the cultivar line that it was using was this cross, when in the gold data set, we only wanted this one. He had, for example, for the disease resistance name, he said that it was resistant to this. And the model outputted resistance to, which make it, when we're analyzing the metrics, match it as wrong because it was not the exact keyword match. So these were small problems that actually made the model have very modest metrics, 0.5, 0.49, which was sad when we look at the metrics at first, but then like looking at the responses, it wasn't that bad. It was a good match. It was just not the controlled vocabulary that we were looking for. And this is, for example, and this is another example in which this happens. Sometimes it was missing, extracting some data, and sometimes it extracted perfectly, but this, for example, it had a different punctuation. In this case, yeah. So some of them were a little bit different, but some of them were actually wrong because it also depended on the chunk that it was extracting. So now we are actually expanding this because that was a prototype that was part of a master's project. And it was a prototype just for the disease resistance thing. So now we're expanding this with Jack and with Dave in a different project, which is led by other Australian partners. And the idea here is to modularize everything that we have. So we have like a pipeline that works, but it is script-based. So the person would have to know how to code well to go through and even like press and do small changes. So Jack here is actually doing a modularization and building everything API-based. So it is much easier to do the processing. And he has well, by now he has all the pipeline for data extraction and processing done. It's beautiful. He's very organized. And then he's also now finishing up the llama extraction pipeline. So all of this should be kind of containerized tool that we can use to then query papers on different questions, because the pipeline is not focused on one question or one species or anything. It's focused on how you want your results to be, what kind of structure you want them to be. So theoretically, it could be used for a range of questions, range of scientific questions. And I think that's basically the update that I've had on how this is progressing. And yeah, just as a summary, like the interest for me is always kind of trying to see the plants from as many angles as possible and trying to leverage the information that we already have, all the scientific information that we already have, to try to describe this and find, for example, new avenues for plant breeders to be able to improve their performance of the plants in the field. And I think the language models it's a great additional data set that we can use to try to build more comprehensive views of the plants. And this is the pipeline that we're developing. And it is mostly, it's a very data scientist focused thing, because it outputs tables and it's a knowledge graph, and you have to have some understanding and some like for data to use it. But I think it can be quite powerful. And that's it from me. Thank you very much. And a special thanks to all the collaborators, funders, and everyone that worked in this project. Thank you.
Questions?
Maybe. Also, can I unmute them maybe? I don't know if they're...
Yeah. So no, it's not. Okay, so online.
Oh, yeah. I don't know.
Hello. Do you hear me?
I can...
Wait, how to do that?
Hi.
Yes, please.
Yes. Sorry, my camera is not working. My apologies. First of all, thank you very much for a very intriguing talk. Interesting results. My question is regarding SHAP value plot. I was wondering if you did any sort of validation for the markers which you found to be like of high impact. Did you have GWAS data where you can find the genotypic variants explained by those markers and also the allele substitution effect, so that this serves as sort of validation and you can be sure, okay, they have biological meaning?
Okay. Yes. It's a great system that we have. Yeah. Thank you so much for the question. So for the SHAP values, we are actually looking now, because like I mentioned, this was a master's program. So she finished her thesis now, and now we had all those results, and now we are actually looking into seeing where those markers are, if there is, if people have published already in the literature, if they have any association with yield, if they're close to any genes or any reported K2Ls and things like that. And we're also looking at differences for them within populations, because it's a very big data set. It doesn't have a very structured population, so they have a lot of crosses that intermix. But we were trying to see also if we have specific markers for a specific portion of the population, and how does that possibly relate to their pedigree. But that's still an exploration at the moment. So we don't know if those markers are, they might be biologically relevant and not have been described before, but we don't know yet how biologically valid and how much we can validate from these results. Yep.
Thank you.
Question?
Yeah, I have a couple of questions. I think the first analysis that you showed was interesting. You use multimodal deep learning using output phenotyping and genomic information to predict yield, and it's pretty high. It's 0.83. It's so high. But is that Pearson correlation? I mean, or is categorical correlation? And what environment was that?
Oh, yeah. So that's a Pearson correlation. And it was, well, the environment actually, I could ask you because the data is from CIMMYT. It's from Obregon. Yeah. Yeah, it was, I didn't have the environmental features, which would have been great to try to categorize. It was 2 years, it was 2017, 2018. And I think they were growing Obregon, something like that.
Yeah.
Yes. But I don't know exactly which environments they were, if there was a drought or anything like that, which would have been, I think, like I didn't put here because they were different species, but I have models a lot with maize from the Genomes to Fields data set, which is also massive, and it runs in the US. And the environmental features usually also provide a lot of, well, it improves the model performance a lot. It helps the model kind of place those individuals, but also it provides a lot of G/E interactions with the SNPs. So we found some SNPs that were correlated, particularly with, for example, this SNP is important for yield prediction if it rains on the 12th week and 11th week of the crop growth. So they were kind of time sensitive, which I thought was quite interesting. And for the maize data set, we actually had time to look a little bit. And that was those weeks specifically where we saw the variation for the feature importance and its correlation with the SNP. People have already described in like agronomy papers how raining or irrigation, if you can, on those weeks were crucial. So that was interesting for the model and for us. Yeah.
I have another question if I can. Yeah, that's a quick question. I mean, how did you assess the accuracy of the Oyama model that you show? I mean, what kind of question, you get it very much, what kind of question I can ask to the LLM?
Oh, yeah.
I ask, I found this gene in this position. Can you tell me if somebody else this? It can show all the papers, right? Is there something like that?
Yeah, so for the first, like for the prototype, for example, we had these questions and we turned them into a structured thing. So for example, the question that they provide to us, which was, I think, the most confusing one for us who don't work with plant pathology, was were the proposed or proven molecular mechanism underpinning the resistance conferred by the R gene? I was like, okay. So we broke that down into different columns because that's what I was kind of looking for, was columns saying, like, for example, if the molecular mechanism was proven, then yes or no. What resistance it was, yes, like the name of the resistance and then the R gene. So for this one, you would have to think of them as which stages of the question. So if you have a complex question, like you mentioned, what were all the genes associated with this under this environment? You have to break down, for example, environments, gene, and then things, and you would provide you a table with that. But this was the prototype, so that's how we initially thought. But now with the work from Jack and Dave and everyone in that group for this other project, we are having a little bit more natural questions, and then the model itself would provide the columns, and then you can have a response, because in this one we're actually using RAG. So instead of processing all the papers like I did for the other, the model will, well, we have a RAG stack in which it finds all the papers and all the chunks of text that were related to your question and provides you just those chunks with the answers. Yeah.
Then we have two. Okay. Quick question about the yield prediction, same site we talked before. So I was wondering if you look at the linear mixed models and how well they performed.
No, we didn't actually, because this data set came from a different paper from Toglianari. I don't remember his name properly, but they did actually another multimodal deep learning model. and they compared it with linear mixed models. So we didn't, we just compared with BGLR, which was also one of the models that they test, which is, I think, a linear regression model, but with Bayesian. I don't know if it's optimizers, but yeah.
Yeah,
but not a mixed linear. Yeah. Yeah.
Machine learning can be better.
Yes. Yeah. No, I took advantage that they had already tested. So I was like, oh, don't have to run that part. But yeah.
Thank you. Jean-Paul, thank you, Ulrika. So now we will have a quick photo together. I will put the people online on the screen. So if you want to appear on the photo, put your camera on. And after this quick photo, we'll have a 10-minute break, and we'll try to fix the the sound. And yeah, so let's start with the photo, please. So I think you can see Tanner's presentation. Yeah, okay.
Yes.
So we'll start in 1 minute. Tanner is giving the attendance sheet to the initiative. You can come in, Tanner. So let's start. We start. Thank you.
All right, I'm glad everything is fixed.
Yes, it will be okay. I put additional microphone. So
Perfect. So hi, my name is Tanner Chen. I'm the lead scientist for the GrainGenes database. So yeah, I work in California, in the United States. Okay, great. So yeah, essentially we are transitioning this year, in the last couple of years, from our main website link with pwusd.gov to graingenes.org. Everything else is still available through the with links, with .pwusd.gov links, but it's actually being forwarded to graingenes.org. There are several reasons why we are doing this, but maybe we can talk offline about that. But our mission remains the same. We are here to serve the researchers, small grains researchers, wheat, barley, rye, and oat. And we are putting new content to our website, new tools, and I will talk a few of them here. So we are a flagship resource for this USDA, and we have data, we have tools, we do research, and we work with our partners. But in the heart of it, it's a small grains community, and wheat plays a big role here. Definitely, without partners, we cannot accomplish anything. AFC is on top of our list, especially for oat. And other consortiums like PanOat, but definitely Tetraploid Wheat Consortium, as well as companies that we worked over the years to be able to bring in data and share the data. We work with groups to make sure that we do have standardizations and nomenclature and data. There is for wheat, most likely most of you are aware of these papers, updated guidelines for gene nomenclature. I hope people are using it. It is cited frequently, but of course, citation is just one criterion here. If the people are using it, then we will be able to increase the fairness of the data, and we will have more easier way to find the data and share the data with others. And most recently, Michael took the initiative to create this chapter for the with data integration. So, and you can find that in the with genome book. We added a new content last year. Creation is a very important aspect of what we are doing. So that's actually one of the things that we are better than some other databases, especially in the United States, because if you're funded through like National Science Foundation, then your grant may run out, and then you just stop doing things. Or if you're creating a new database, then you create some tools, maybe genomic tools, and they may be out very quickly. But putting genetics data sets is not as easy. And we have been doing this since for over, I don't know, 1990. We are around since 1992. So we have been doing this for a while now. I guess I shouldn't have ran from the foyer to here. All right, sorry about that. Okay, so this year we added some protocols. So MaxWheat protocols, for example, come from Jorge Lipkowski. So Jorge was leading the WheatCap project. So we brought a bunch of protocols from his lab into GrainGenes. We also have some protocols shared by Isabel Fassi Gomez and Cristobal Luoi. And I should say that that's putting the protocols in the in resources is really helpful for other researchers. But after putting the the protocols, Cristobal became the director of John Innes Center. So it has direct. I'm just saying. If there's correlation, is causation, right? So it's like definitely helps putting your data into databases. So we do have new tools and use. We keep adding new genome browsers and BLAST. We have a very rich set of genomes and annotations. Our data sets are becoming more and more pangenomic. We do have barley and wheat pangenomes put recently, and Silver2 is available, was available under the Toronto Agreement, but now it is available with open access. So for pangenomes, we do have two different views. One is JBrowse 2 base, so you can see the comparative views. I think these tools are great, but with JBrowse 2, JBrowse 1, and on ensemble browsers, it's the same way. It gives you, in a sense, some two-dimensional views, yes, but it's more like one-dimensional, especially if you're looking for single genomes and annotations. I think there's still tons of space for improvement. So and especially when now that pangenomes are becoming more and more available and easy to generate, I think we need to think more about how we can visualize them better. There are some ways of doing that. JBrowse provides this comparative views. There are some graph pangenomes, but they are very local ways of looking at it. So there's definitely computational cost to it. I think this will be for people looking for like how to visualize things, or if there's a problem, this is a problem that maybe they can start looking at. Yeah, if you're lucky. For the genome browsers, I'm a big fan of with other people as well, linking so that a user can come to one place like our database, and then can go to other databases. So for the wheat, definitely WheatIS is a great place because we can actually, a user can come and then go to another website to get more information or to use the tools there. So I'm not talking about a database level linking, but the interface level thinking, because the user interacts with the interface, not necessarily the database, right? So that's why we try to give links, bunch of links from different browsers to different database resources. T3, the 3TC Toolbox, is a very close collaborator. We usually look at the raw process data for genomes, and T3 focuses on genotype and phenotype relationships. So we work closely with them to provide this linking. So genomes are getting easier to sequence, assembled, but the genetics somehow we are a little bit behind when it comes to genetics. Luckily, some groups pay more attention to like QTL and GWAS data, and we try to put these as tracks on genome browsers so that you can click and then you can go to the QTL pages and you can get more information about this QTL. So that you can go, and then from the QTL page, you can go to the genome browser. So this is essentially providing an opportunity to go from genetic data to genomic data back and forth. And that's, I think, important because as user, sometimes you want to start at SNP, you want to go to genome browser, right? And then you want to go to some marker data, later other types of marker data or QTL data. So I think that a good interface should provide this dexterity, this capability to users. And I'm not saying that we are doing a great job of it, but we are striving to provide these kind of things. Where am I with the time? I'm too. Am I good? Okay. This is why you gave me more twice as much time that I asked, right? And you're like, hey, Tanner will just talk about stuff. Great. Yeah, we do have the blast for nucleotides and protein. So you can visit our website if you're not familiar with it. So something different than other databases. So you can actually, if you have a sequence, you can blast that, and then you can actually go using the links. You can go to the genome browsers we have, and then that region is shown in yellow. And then you can actually choose. I don't know if I put it here. Let me go. Yes. You can choose an area in the genome browser, and then the sequence is transferred, peer-populated into our BLAST interface, and then you can choose the database you want. And then you can just essentially come back and forth, going back to that linking that I talked previously. Recently, we put this AlphaFold links as well. So for some genome browsers, so that you can click, we create a track so that you know which proteins structures are available, and then you can go to the Ensemble website to see what that protein looks like. Other new tools we added. One is FoldSeek. So for different proteins, it provides the way that structurally similar proteins than the one you have. These are all predicted structures. Of course, AlphaFold is very powerful. At least for this is AlphaFold 2, I believe, although it's called AlphaFold. It is very good for certain structures. It fails miserably in others. But if you're lucky, then you get a good structure. For example, this is a good structure that we see here. But in some structures, you see tons of disordered regions. And this may be because of the lack of data. It may be the failure of the algorithm. Or sometimes we see that there are some chains that people put in, but then there's a difference between proteins in biological sense versus the crystal unit proteins and how they interact, so that you may have a monomer versus a dimer or vice versa. And those things are not always captured using AlphaFold 2. So you should be aware of the biologically how that protein appears in the search you have, and the associated sequence information. And also we have another tool called Synversity. So here you can have a reference genome, and then we can show the variances against that genome, so that you can go to different. It's a very colorful view, and then you can look at where the differences are, and then go to the genome browser from there. We also collaborated with other groups. This is for actually for Fusarium, Pangenome. Here you can see that the differences in the protein sequences and how that mutation possibly affect the structure. So this is just for the coding regions. But at least for the Fusarium case, it can show you what an effect may be. Usually variations in proteins are, I mean, actually maybe not, it can be deleterious, right? If you have, for example, polar to hydrophobic transition, then your protein may lose its stability. So it's I think it's useful to see those kind of differences in a view that will allow to see that will provide you further insights into building hypotheses. So we have that for the what we call the Fusarium protein toolkit. We also did research in the recent years. So this is about the PFAM domains. PFAM domains are very, as you know, is very useful, and we all use it. But they are usually 6 sequence based, right? So we thought that, okay, we had these families, and the PFAM essentially created these families. So what if like we do a structure prediction and then see what they look like? Of course, these are not always the whole protein, but part of the protein. So in this case, domains may not mean a single globular part of domain, but it may mean that a certain region of the protein. So it's a distinction that is, I think, important. But but we thought that we'll see very similar structures. And after we did the clustering, we see that there are actually some distinctions. So you can see the principal component here. I don't know if it's easy to see my mouse, but at least in this case, okay, there is some difference. But these proteins 1 and 2, this I and II, essentially, it can look okay similar. There are some alpha helices, there are some beta sheets, but we did have some proteins that in the same family we can see a coiled region, not the whole thing is coiled, or only some alpha helices formed or beta sheets formed. So we do not expect to see this kind of variety in the same PFAM family, in the same PFAM domain. So so it was very interesting for us to see that. And one of our reviewers was Alex Bateman, and he was happy about this to see that there are some ways of improve PFAM. And actually, he changed some of the PFAM assignments based on our research, based on the paper, which was a great experience. You know, sometimes you publish a paper, you don't know how many people will read this paper. So in this case, there was some direct effect, and it was good to see that things can change because of your research. We also did we look at whether we can use the large language models in curation, and we try to compare this AI bot versus the PhD level human curation. So we try to do a few things. We try to get the PDFs, and then this is like for 30 plus papers, and I think we can do much more right now. But we use GPT-4 3. Let me go to the next slide. 3.5 and 4. I mean, right now you can have GPT-5 and others. So we look at the PDF and see if we can use some interactive module to be able to extract data for QTL and MTAs. So yeah, actually we were very surprised that LLM were very good at extracting data when compared to humans. Maybe I can show this. I mean, we try to, so this AI shows the prompts, and then we learn very quickly that prompts are very important, right? So I think it's a learning process in the sense that, you know, maybe some of you will remember it used to be that, oh, how can I ask this question to get this information? And sometimes people say, hey, you have to ask it this way. And then Google search came and searching was much more important, was much easy. And now we are at this stage, we are asking ourselves how we can ask the question, the prompts right, in the right way to be able to get the best results. And that's especially with the bots, the hallucination is a big problem. It just it can just lie to you with straight face, let's say, right? And you have to definitely prompt this, saying, "Hey, don't lie to me," or if you don't know something, you have to tell me, type of prompting, and still you can fail, right? We try to actually trick the program by asking peduncle lengths. It confused a bit the panicle length and so forth, but especially with you can see the G there, GPT 3.5, the confusion was very high, but then GPT-4 just fixed tons of problems, so which is very good to see. So I'm sure GPT-5 now is doing much better. And we saw this overall, going from GPT 3.5 model to 4, just enhanced the results. So we play with the chunk size and top case size, etc. But yeah, it was very good. We also try to see whether we can actually get better results by asking the questions a certain way. So here we do have in A the black part, and then this orange part that gives instructions. It's extra, hey, to do this, just essentially giving guidance. And then the underlying part is another thing we change, essentially we remove or include. Like, hey, a range, I'm looking for a QTL and make sure that the range is this, chromosome 2B. And when you ask questions in a more detailed manner like this, you get much better results. So it is better to ask the questions. If you know where that QTL is, say this is where it is. Just tell me that, look at chromosome 2B. So this actually is very good to tell people, especially biologists, because in our experience, biologists know their regions. That they know which genes that you can find in that region. So if you're more precise in your questions, then you can get better results. So yes, definitely AI versus curator. Humans that time are still much better at this, but definitely GPT was catching up, and it is very comparable, and it's getting better in extracting data. So yeah, this is our experience. And now there are actually companies that you can feed the PDFs and get tons of data out of it, tons of extraction. So I think a better study will have include these companies and what they can actually provide you in the end with respect to what you can do in-house. Because we are using AI, but very from a genetic sense. We are not computer scientists. One of the things that the AlphaFold did differently, there were like for decades there was the CASP experiments, the community experiment for protein structure determination, and I don't know how many of you are aware of CASP actually. Okay, so okay, it is Yeah, once upon a time it was very important. Every 2 years there was this experiment where the X-ray people determined the protein structure and did not tell the computational people what they were. And the computational people tried to predict these structures and then just compare them, right? In the end. So, but things changed when with AlphaFold, because AlphaFold just killed the competition really. And afterwards, there is no CASP anymore, essentially. Okay. But the important thing was this. So AlphaFold did not just like start doing stuff. AlphaFold based everything what people have done, there's all these methods like for decades, it used that, right? And on top of it, they use cutting edge computer technologies with the neural nets, essentially. So it was not, so they used the biology, computational work, and computer science knowledge. So very specific way of doing collaborations and creating these tools. So I think going forward, this will be the model. You will have the computer scientists knowing the models much better than biologists and maybe computational biologists. But you can do only so much, right? And the pace is very fast right now with the large language models and AI. So we'll see what happens in a couple of years. I mean, I'm aware of some they're trying to do AI scientist. I don't know if you see the code scientist paper that was just kind of published. The preprint was published through either Science or Nature a couple of days ago. They are trying to use create AI that can actually help out the scientists doing experiments, creating this hypothesis, a new hypothesis. So we'll see where things will be in a couple of years in that respect. So yeah, we do have some new tools, but I don't have time. But so yes, it's great to be here, and thank you. Yeah.
Thank you. Very nice presentation. I saw a lot of data to index in WheatIS. About what you said about the pangenomics and so on, the visualization, we have the same, we think the same thing, and we are trying to have some synteny viewer using graphs between genes. So perhaps when it will be finished, we can look at it.
Actually, we are developing this new views right now so I think it is important. So the solution we found, instead of looking at everything, for wheat, we look at the orthologous regions, orthogonal genes, and then build these views based on those genes. So yeah.
I think we have to share some.
I can show you a few afterwards. But I think then you can see. So it's more like a gene base, so that but you already pre-calculate the orthologs, and then think about the synthetic region. Then you can just go and then see what that region looks like on a either genome browser, or much better, I think, to see a view where you see the genes as they are, and then you can actually adjust the regions, and then you click and then go to genome browser afterwards.
Okay.
I can show it. Yes.
And about the curation, do you think in the future that the AI will be assistant to a human curator, or it will be only AI and no more human validation?
Yes, I don't think anybody knows the answer to that question. We hope that the AI will be helpful, but even the jobs of curators are very precarious, right? Usually grants do not support curators, and it is very difficult to find the right curators doing 100% of the time curation, right? I'm asking this question myself. I'm sure tons of people are. I see that. Sometimes when you create a website, as you know, right, you have to find a programmer. Programmer works with other people to create this website. But now, for example, my postdoc just created this website initially using just the tools, AI tools, scripting tools, very easily. Then, I mean, you need someone still, it's very clunky, right? So you have to go through it. But the initial website was up.
It can get the time.
Right. So seeing that curated work, now there are some companies you can just put the paper in and then you get some output. But with everything else, I think so you have to look at the workflow. So I think that a single step, single job, like, oh, give me the PDF and then here's the extraction, are done easily. But how to connect them is kind of problematic right now. Somebody who's an expert needs to go through it and then just do these steps by itself. I think that it's very similar to, for example, if you want to go to a vacation, you don't know the days, maybe you're flexible with the days. There are flight information, there's hotel information, and sometimes you can just get help for each step, but the whole thing is not done yet very nicely. So I think it's the same problem. Create you have a data component, the AI can handle some of this. You have a programming component, you have the analysis component, and then you can use different AI tools to do different these tasks. But I think at one point, and that one point is not 20 years from now, it is maybe 5-10 years, that there will be programmers, there will be some tools that will allow you to do the whole thing as a workflow.
And so you will only review
a review or something. So
yes, I've done that for live fiction. So already AI planning, full AI planning. We discussed that.
Well, there was software. It's called AI Professor and AI Student. Okay, so the same company was creating these two different apps. So AI Student was interacting with Canvas. Canvas is a very popular software. I don't know, in the US at least. So it can interact with the professor, like asking questions to professor about stuff, writing emails and everything. But then company was actually providing this AI professor on the other side to sell to professors how you interact with Canvas and how you actually communicate with the students. So essentially AI bots were interacting with other AI bots in this case. Yeah.
So questions, one question and then you can go.
Sorry, I have a rambling style, so I know that. Sorry about that. Okay.
I'll let you know how my holiday turns out. My AI generated holiday in a couple of weeks. How much hallucination I got from me. But my question was, I noticed when you started your presentation, you had agelab Starship in as a display, which makes sense because it's the genome of wheat. But you also had agelab Mutica. What's the interest in grain genes in populating with all the agelabs? All of them.
Yes.
So 25 of them, because I got the data.
Yes, please send us. Yes, yes.
So what I was asking is, if we put the data into GenBanks, once it's published and it's there, can you extract it from there, or do you need different formats? So that's one. And two, you talked about pangenome issues, but when we're talking now about how do you see not comparing the same genomes across different varieties or genotypes, but visualizing comparing different species altogether. So that's question.
Yes.
Where you can get the data,
right?
And the format, and two is the sort of visualization vision, if you wish.
Yes. So first of all, we are very open to acquiring data. AgileLabs is very important for wheat researchers, and we have also other wild wheat as well. So yes, please send us the data. You can say, hey, our data is here, our is there. We just need the FASTA files of pseudomolecules. at this point we only accept high quality, which is the case, I'm sure, with your NCBI submitted data. Let us know. Annotations, if you have annotations, send us a JFF file. It's much easier. We'll just put it up for you. And yes, EduLabs is very important for us. About the pangenomes, so is this a question about which pangenomes, which genome assemblies to actually assemble, or is it how we can go from one genome assembly to another?
More of a question, what's your vision and visualization of what one can compare? So in a pangenome, you're usually comparing different varieties or genotypes from the same species, but is there a vision or possibilities of comparing different species?
Right. So yeah, it's very difficult, right? There's so many challenges. So once upon a time, we had this one reference genome that we can actually use against, right? We can use the variants and everything. So, and as I said, that graph pangenomes can provide, if you look at locally, then it can give you some ideas. But I mean, these are some solutions, not the perfect solution. So this is why I think if maybe they're looking at the gene level, synthetic genes, perhaps it may work. But again, it's not a perfect solution. But this is what we found out, that with the pangenomes, that's the best solution we have. Instead of looking at regions, just look at the genes that are orthologous and then go from there. Still, there are questions like so sometimes you have triads, right? Perfect for the wheat part, right? But then tandem genes, how would you handle tandem genes? Copy numbers there. So there are all these questions. So yes, you have to start somewhere, but then you have to look at your region and then go from there to the genome browser, genomic region, and then see the annotations there. But in the end, you still need to go and then look at things locally. You know, for example, gliadin genes are not very well annotated, right? And that's understatement right there. So how would you do this? People who are doing the genome computations, we pay attention to these specific gene families when we need to look deeper. But usually these annotations are very general, So these are all. So this is why they are really called gene models. Some models, and they're you need to improve them. I can show you just like a. Do you have like a one? Oh, okay. Yes, but thanks.
But you can interact after.
Yes.
So because after Paolo talk, we have the Q&A presentation, and we have to finish in
yes, I understand.
half an hour.
Sorry about that.
No worry, it was very interesting.
Yeah, I'll try to be quick. So, well, I'm Paolo Vitale, quantitative geneticist working at CIMMYT in the bread wheat program. And basically today I split the presentation in two parts. There's the first part where I will describe our quantitative genetics application that we carry out in the bread wheat program. And another part is basically our data management system that we are currently using at CIMMYT, in CIMMYT. So about the first part, this is basically I can use the laser, right? No.
It doesn't. No, you
doesn't work. No, I can use the yeah. So basically, this is the general pipeline. As you can see on the top left, we started supporting in the crossing block selection. After that, the parental selection, followed by cross prediction. From F1 to F3, we cannot do too much in terms of quantitative genetics, since we are still at family level.
And F4, there is the high throughput phenotyping application. At F5, we have the genomic selection. And from F5 to X6, we can have sparse testing. We didn't use it yet. And then there is the GBV's estimation that we can use to close the cycle, basically. For the crossing block selection, first of all, what is a crossing block? Crossing block is a group of individuals that the breeder select as candidate parents, okay? And in this case, we adjust the phenotypic values that we collect in the field using the markers. In this way, what we are doing? We are improving the precision of that phenotypes that we collect. Okay, improving the precision is useful because it's directly linked to the genetic gain. If we can see the phenotype more accurate, we can have a higher genetic gain, basically. About the parental selection, so far we used 3 methods. I will not explain each of those in details, but we can say that all of these methods basically has one name: optimize the genetic gain. Basically, these methods are going to select those group of parents that crossed randomly among them is going to optimize the gain. We have the Bayesian decision theory, the identification of parental RNA and genomic selection, and the desired genetic gain index. Okay, this is not, how can I say it? It's not the best things for the breeders, because every time that I deliver the result to them, they always replay, okay, but we already know that they were the best in the line, so can you give us something more? Well, something more is basically how to cross these parents, these candidate parents, right? Because okay, they can easily identify the best ones, but how to cross them? This is the key part. And in this context, cross prediction might be very useful for them because basically we use a software that is called PopFar, where basically in a biparental cross. There is in biparental cross, there is the simulated offspring. We can simulate the offspring of the biparental cross and predict using genomic selection the average performance of that cross, the additive genetic variance of that cross, the top lines of that specific cross, and this is usually is a key information for the breeders that can select their cross using this criteria. So after that, as I said, from F1 to F3, it's hard to intervene. I mean, we are still familiar. At F4, we basically are in a stage that is called head roads. Head roads are basically small plots. At CIMMYT, we are, 25K lines for pipeline. We have 4 pipelines, so we have approximately 100K small plots. In this case, we cannot phenotype conventionally, too many. We cannot genotype, there are too many. What we can do is basically to fly a drone, collect a reflectance value for each of these plots, use this information along with the pedigree data, which is always precious and available, and we can predict the target trait, which is basically always grain yield, okay? A breeder can use this information to discard the Ross lines or to select parents. If they can use this technology to select the parents, they can basically close the cycle 1 year earlier, and which is fundamental for the genetic gain. Indeed, basically this is the genetic gain equation. Basically, you have the intensity of the selection, the accuracy, the square root of the added genetic variance, and the L terms, which is the length of the breeding cycle. If we apply this, we basically are gaining in selection intensity because we are selecting less individuals. We are gaining in accuracy because we can see the phenotype way better of the so-called breeding eye, because at that stage, breeder, the only things that they have is the breeding eye, okay? And of course, we are going to decrease the square root of the added genetic varies because we are doing selection, and we are decreasing the cycle. Decreasing the cycle is a key here, okay? This is just a cross validation to show you how much we can predict. I mean, how much well we can predict the target rate. In this case, is grain yield using a marker-based model and phenomic-based model. As you can see, when we add the phenomic-based information, we can improve quite a lot the prediction accuracy. Of course, there is always a bias here, right? Because when we predict using the markers, what we are predicting? We are predicting basically the genetic merit, right? We are predicting the additive values of the lines based on the markers. When we train in the model with phenomic information, we are adding also the environment. We are training the model with the phenotype, right? Which basically the genotype plus the environment. So it's normal to have a predictability higher, right? It's logic. But it doesn't mean that this improvement in predictability is an improvement in prediction accuracy, which is the term that we have here in the genetic gain equation. Kind of complicated, right? Well, this is a couple of numbers, a timeline for our genomic selection pipeline. Basically, we start with approximately 250 crosses. Then we have 250 families, so of course, unless we discard a few families in the way. And then we have an effort approximately to 25K lines. And after that, in the F5, we are basically have 3.7 lines. After the screening, we have 1.5K lines, and these 1.5K lines are genotyped, and basically it's our target population. Okay, where we are going to predict for all the target traits and deliver to the breeders the prediction that they use for selection scope. These are the traits that we annually predict in CIMMYT. Basically, there are grain yield in 6 selection environments. There are several disease resistance, yellow rust, stem rust, etc. There are zinc and iron, and there are a bunch of quality traits. Basically, we use our historical data as training population, and the target population, as I mentioned, is this pre-yield trial, this 1.K, but it's by 4, which we should be 6. And yeah, it's 6 exactly. And the breeders, basically, we provide the breeders the GBVs, then they use the GBVs to make selection. Should we use all the data that we have? Well, we have more than 10 years data. We basically have seen that it's not actually useful for the model that we use to use all the data. Because, of course, the relationship between the target individuals and individuals of 10 years back is very small. They don't have almost any relationship. So include those lines in the models basically is not useful. On top, also the environmental conditions are changing, right? We are targeting a new environment, which is probably very far from 10 or 20 years ago. These are two key points. And we identified that basically a plateau within 4 and 6 years back of training population. This is the model that we use. Basically, it's a GBLAP model. We sometimes we do GBLAP and reproducing kernel Hilbert space, but the structure of the model is basically the same. You have the intercept, the effect of the environment, the effect of the genotypes, which include the genomic relationship matrix, the G by E effect, and the error. How we affect the genetic gain equation by using genomic selection. Of course, the selection intensity is increasing because we are selecting less lines. The precision is increasing as well. The square root of the added genetic variance is decreasing because we are doing selection. But the breeding cycle, in the way that we are using a genomic selection sim, is not very. So basically, we don't have a huge impact for the genetic gain by using genomic selection as we do. Of course, there are other implications, right? Because it's not just that step. We can leverage the genotyping, for example, increasing the precision of our stage 1, stage 2, and stage 3 lines. We can save a plot using sparse phenotyping, and we can use an optimized cross selection, which is basically the process that I mentioned before. So a couple of slides I asked to our head of the data management unit to provide me a couple of slides on our data management system, which is called Enterprise Breeding System, EBS. So she provided me this slide that I'm going to show you. Basically, in the EBS, so far there are germplasm data, phenotypic data, and the genotypic data still is work in progress, let's say. The system is ongoing from the April 2022. And basically, it's also linked to our germplasm bank. Talking about wheat, of course, I think it's more than 100,000 genotypes, including old accession and new lines. The aim of EBS is basically enable more efficient workflow, facilitate the work between different teams. Of course, it's easy with EBS to share data. Because I mean, I'm, for example, in the biometric groups, biometric quantitative genetics group. I don't have to ask the breeders to send me the data. The breeder has just to upload the data on EBS, and I can easily download from EBS itself. Then also to promote high quality management of the germplasm and data. What kind of data are right now in EBS? Data from the bred wheat breeding, durum wheat breeding, molecular breeding, pathology, physiology, international nurseries, quality data, seed health laboratory, germplasm bank, as I mentioned, biometrics and quantitative genetics, the unit where I belong, and data management too. A couple of numbers that we have here. Basically, there are more than 9 million of germplasm records, 3 million of inventory records, 15K experiments, and 5 million of observation, and the genotypic information, for example, for the GBS, talking about the breeding materials that we have on the germplasm bank. For the GBS technology, we have approximately 64 samples genotyping using GPS technology. We recently switched technology. We are now using Dart tag technology, which basically it's different and hard to combine, I guess. The connections. I think that this is a strong point of EBS. which is basically connected with the Fieldbook, which is the app that the technician used to collect data in the field. So basically, the data are directly stored, directly collected in EBS through Fieldbook. Cloverfeed, which is the application by IFEN for image analysis. They basically linked this application to EBS as well for our trials, of course. BioFlow, it's an analytics software that the CGR group is working on. Basically, it's allow breeder to perform a lot of analysis, including trial analysis, single trial, multi-trial analysis, parental selection, optimal contribution selection, and genomic selection and GWAS as well. So basically, the user can use trials from EBS and import the trials in BioFlow and make their analysis. Fairground, which is pretty new, and I don't have any detailed information on this, but it's something that the Gates Foundation is pushing, and it's basically it's like a data lake where basically our CGR center is putting a lot of data in that. All types of data, including markers, including multi-omics data, including high-throughput phenotyping with trial analysis. I don't know and then it's powered by also large language models as well. Dataverse, as I was mentioned, I mean, I mentioned to you, Michael, because I mean, I'm pretty new in this group, so I mean, I have limited knowledge on the big picture. So it's linked to WheatIS information system as well, where we basically in Dataverse we basically include the international WheatIS trials and or basically the data that we publish. If we made the publication, we used some our stage 1, stage 2 trials, for example. This data are on our Dataverse as well. Yeah, it's basically the Dataverse thing here. A couple of acknowledgement. Leonardo, which was basically the main breeder, is not with us anymore, unfortunately. But now the main breeders at CIMMYT are Guillermo and Bello that are in this conference. Kit is the head of the data science group at CIMMYT, which included the data management and the quantitative genetics group. Jose Crossa, which is always with us, making a lot of suggestions. Susanne, which is the molecular breeder, and Kate, who provided me the slide, which is the head of the data management group. And thanks a lot for the attention. What are any questions?
Yeah, thank you, Paulo. Yes, we at the time we indexed two things. There is two databases, one with the literature and one with the data sets. So, yeah.
Okay, there is a link.
Yeah. Yes,
yeah, because
I think the fairgrounds data lake stuff seems very interesting. Perhaps another time.
Yeah, we can.
Be interesting to have a focus on that and what you can talk.
Michael, I also mentioned to Kate the possibility of, I don't have the cursor here, but the possibility to link EBS directly with initiative. I think it might be something useful, but probably with some authorization. I don't know if this is possible, but it's something that we might need to discuss.
Yeah, but as I understand, the INRAE is more the database part, So I don't know if there is an interface
Yeah.
connected. Yes, there is one.
There is an interface where user can basically check and see all the trials that we have, which basically a lot of trials. It's including years of trials, but the breeding ones, not the international nursery.
Yeah, I think we have to discuss, because in the WheatIS data discovery tool, we index only open data, and
Yeah,
in
no.
eBase you also
We are
have
not, yeah.
internal things.
Yeah.
So yes, I think we have to discuss.
Yeah, we can discuss about.
Is there any question, Monica?
Thank you so much for your talk. I have a question about the project that you presented before with the GBLOBs and the simulation and everything. I was wondering if you tried using different prediction models, because I know, for example, Montesino Lopez group that I think works with Crossa uses also a lot of machine learning and different types of methods for predictions.
Yeah. Of course, we did a lot of tests. I mean, I spent a lot of time doing tests. But in this specific prediction task, the machine learning and deep learning model didn't apport too much. They were like at the same level of GBLAB. And since it's the same level, I usually prefer GBLAB or the reproducing kernel Hilbert space. But in any case, it's difficult to get some gains. But in these prediction tasks, where we basically have several years of training population, and then we have an unknown year that it should be predicted. In this prediction tasks, we didn't see any improvement. But I mean, it's just a specific prediction track. For example, in other prediction tasks, also, as you mentioned from the Tonignali, the data that you use from Tonignali, they have seen a lot of improvement, but different data, different prediction tasks, it's a different story.
I think if you only have, for example, if you're using mostly table data, for us also, the deep learning things works better if you have images or other things that are like a different type of data input.
Yeah.
I also had another question about because you have many years of data, and imagine with those, there are different lines and different hybrids and crosses in each one of those.
Lines.
Do you segregate them by environment or climate? For example, like to do like a climate analog analysis to know where you're predicting from what you have?
Yeah, I mean, unluckily, we don't have the climate data for the future. I mean, it's something that we have think about that. We talked about that, right? But no, we didn't check it yet. In any case, we use a single trait approach where different years, we treat the years as the environment, and we can leverage the genotype by year effect in our models. But yeah, it would be interesting to modeling the environmental covariables, but this is the key, that we don't have the environmental covariable for the future. We might to forecast those. This might be an idea. Yeah, we talked about it.
Okay, thank you. We'll have to move forward. Thank you.
Thanks a lot.
Okay, now we give the mic. You have a question, Mohamed. Okay, you can do it.
Yes, thank you. A very quick question regarding high throughput phenotyping. My question is, are you also using hyperspectral sensors, or is it only RGB and multispectral cameras?
Yeah, basically it's can you hear me?
Yes.
Yeah, I think, yeah.
Okay, you can hear me. No, basically it's multispectral data. In multispectral data, we can collect 5 different bands plus all the vegetative indices, and we can use this information to make a relationship matrix basically. And we use this relationship matrix as a genomic relationship matrix, and so basically we use this to make prediction.
Okay, perfect. Thank you.
No problem.
Okay, Kiwen, you can share your screen.
Can you give me permission, Michael?
I think you are. I know I have to cancel mine. Okay, now I think it will be okay.
Let me check. Oh yeah, now it's letting me. Okay, just one sec.
Yeah, thank you.
How much time do we have, Michael?
In theory, 10 minutes. But yes, if we are 5 minutes late, it's not. There will be enough food, I think. Sylvie, it's our limit for 12:30 or no?
No idea.
Okay, so you
What
can
is it?
take a bit more.
A
Sorry,
bit, okay.
we are late.
Yeah, well, I had prepared for like a 25-minute session, but I can try to drop things and stick to 10-15 minutes. Keep it brief if you want.
No, let's go.
Okay. Right, so quickly start with introducing myself. So I'm Kevan. I'm founder of AI-assisted gene platform called Netminer. It's mostly used for gene discovery. I'd just like to spend a couple of minutes showing you what Netminer is, where we are, and then discuss ideas for an AI hackathon that hopefully make more sense once you've seen this. Right, so what we do at Netminer is we are building knowledge graphs for genes in crops. And this is an example of a gene knowledge graph for wheat. So here's a wheat-specific gene. And the type of information that we are collecting for a gene could be, for example, is a gene present or absent in specific wheat varieties? Is a gene co-expressed? And what is that co-expression module doing? Over here, we've got a gene linked to headwax, and that relationship is coming from a GWAS data set. So headwax is associated with a specific SNP based on this p-value, and this SNP is in close proximity to this gene. And then over here at the top, I don't know if you can see my mouse or not, but I can select stuff. Over here, we've got homology information for different genes. Over here, we've got text mining information for genes. So we can see a gene is regulated by another gene that is published in this paper. And this paper talks about lateral root number related traits. Hope that makes sense. So this is an example for one gene. We have similar data now for every gene in wheat and across crops as well. So the challenge is now, how do we build? I'm not going to talk too much about how we're building this knowledge graph. So a lot of it is automated. I'm going to talk mostly about how can we search this data efficiently. And what Netminer is really a powerful search engine that lets you search across knowledge graphs. And the search engine looks a bit like this currently. It's a fairly traditional sort of search where you need to know where to click, what to put into the different boxes to do a really good search. And we spend a lot of time trying to make it user-friendly. Still, I'm getting first-time users who come to the platform, plant breeders, for example, and they have specific genes in mind. They go into the wrong search, put it in here instead of going to gene list search and putting their gene IDs here. They do not know which gene IDs to use to put into those boxes. So we went and prepared lots of examples to give people an idea of, okay, so if you're interested in wheat, put your gene IDs over here, your search terms over here, click a search. But it needs a lot of training, onboarding to make people just use the platform the way I would use it, or you guys would use Grain Genes, Ensemble, and all the other platforms because you are experts in using those platforms. But for new users, it's hard. So with that in mind, we started thinking about a natural language interface into the knowledge graphs that makes it all a bit more intuitive to use. So I'm going to switch now to something we've been currently working on. It's the natural language interface. And this is now, looks a bit like a normal chatbot, but the important bit is it's working on those knowledge graphs. It's not going to use it's going to combine its own foundational knowledge plus all the data it can retrieve from Netminer to reason over it. And what I can do is, for example, I can show you a couple of examples. So there's a paper here that I was just looking at. This is a publication in Plant Biotechnology, published just a few days ago, 2026. And if I go and download the PDF, go into, where is it gone? Go into the search, upload that PDF. Can you still see my screen?
Yes.
You can. Okay, so now
we cannot see the mouse, but we see the screen. So
okay, so I uploaded a PDF. It just prompted me to upload a PDF. So I've taken that PDF, and now I can go and ask a question like, which wheat genes are mentioned in this paper? It's very brave to do a live demo, and yesterday evening it wasn't working. I had to ping the developer, say, guys, I'm running a demo Sunday morning. Need this to be fixed. And they actually got up early and tried to make it work. So it's early days for building this, so it's not very stable. But you can see, so it's using its own, just a normal AI capabilities to scan through the full text paper and identify the genes that are mentioned there. So here we go. So lots of gene names mentioned. And now I want to carry on an analysis. So suggesting, so do you want to search these genes in cereals premium? What do you want to do with them? Can you find the gene IDs? What do we say? The matching gene IDs using plant slide. So plant slide is a knowledge graph that is public and free. And so we wanted to go take those names and see if it can find the equivalent gene identifiers, because that's normally a first step where people, will start with a paper, they read all those names, but they don't necessarily know, okay, what are the identifiers I need to use now in Ensemble, Netminer, grain genes, maybe. Okay, so it's now using the Netminer knowledge graph and I talk about it, some skills and that it has to map names to identifiers. And then I can carry on now that I've got identifiers and it has grouped the genes, I can go and ask the next question. And for example, which traits are the RTV, what are they called? RTV1 genes linked to? I think one thing that's really important to me, especially as while we're developing this, I want to be able to trust the system. And to trust it, I want to see the exact API calls it's making and the data it's retrieving. So what we are trying to offer is not just its own reasoning over here and the response, but those green boxes that you see here, if you expand them, it shows you what so Netminer has various of APIs. It's calling now the search API with these parameters. It did understand the question. It managed to parse it, translate it into a structure that our API understands. And this is the result that it has received from the API. It's able to search through that and then grounds its answer based on that data that it has retrieved. Does that make sense?
Yes, I think the people in the room cannot read exactly, but it's saying loading evidences per gene and explaining what he's doing.
Correct. Okay, so there's just one flow, right? And then flows could look, now we've got this natural language way of searching. The flows could be, we started with a paper. How about we started with a CSV file, a GWAS output? Let's say someone has run a gap hit and wants to analyze the significant SNPs in here. So I've uploaded a GWAS file, and then can I ask it, so which here's a GWAS. Let's explain it. Here is this is Arabidopsis GWAS, Arabidopsis GWAS. which genes are linked to the most significant. Ooh. It's okay to have typos, right? It will be smart enough. So now it's a simple file as in SNP locations and p-values. So it needs to understand that it needs to do some filtering, identify the significant p-values. I didn't say what my p-value threshold is. It might ask me that question, or I may need to clarify it later on. And then it needs to find windows around those SNPs and go and fetch genes that are in those regions or automatically. So it needs to plan those steps, understand it, know what skills to call, and then go retrieve the data. And once it has done that bit, it should bring me back to the classical netminer and show me a network or show me a table of the outputs it has generated. So it's learning. It's learning to do those tasks. There are various flows that it can already support, but there are still lots of things that it hasn't doesn't know how to do exactly, but it has the potential to be taught how to do those things. Now it might have generated something that's far too big and it might crash the browser. So I think really what is key is not the AI bit of it, is maybe 2%, is the 98% around it is the setting the guardrails, teaching it when to retry, when to stop, is the software engineering that goes around it. That is far more important than just calling an AI chatbot to get some answers back, right? I think, yeah, this is probably not going to finish now and might, crash the browser. I don't know what's going to happen. So if I just reload and show you just one thing. So I think what I want to discuss here for the AI hackathon could be a topic of, now this natural language interface knows about netminer data. It can connect to an endpoint we call MCP endpoint that has specific skills, but it can equally connect to other things. And so there are public connectors, for example, BioMCP, that it can connect to, and it can get or search UniProt, Cambo, open targets. A lot of these databases are biomedical clinical trials. There's barely anything that is plant specific, and definitely nothing that is wheat specific. And I'll just show you a registry that the group in Munich is developing around BioChatter and BioCipher. I think, Michael, you know them. They're building this registry called Biocontext AI, where you can register different endpoints, and they would give you, if the endpoint is public and hosted, you can copy this MCP connection to the endpoint, and then I can go and add this to the connector here. I can say connected to Biocontext test connection. So now it connects to that endpoint, and then MCP is a bit like an API, but tuned towards AI model. So you can see there are 49 tools and skills that this specific Biocontext API offers. And it can, now if I ask specific questions that require it to go to PubMed Central, PMC, to get full text, it would use, for example, this connection to go and get the data. So would it not be nice, I was thinking, to have something that is like a WheatIS connector, and it has the knowledge of all the different databases we are working, the different use cases that we are interested in. And then it can be either connected to netminer, or you can connect this MCPs to any other platform, right? It doesn't need to be netminer. Once they're registered in this Biocontext AI, anyone who is interested in building agentic flows could come and grab the endpoint and connect it to their toolkit, for example. That could be an idea of something to work on. I'll have a couple of...
It will be useful also for people which are not in the group and they just want to add the wheat data to their chatbot capability.
It's very interesting thing. Comments on the room? Tanner, what do you think?
Yeah. So I think there are also acquired data is also has some working groups. K1, maybe if you wanna join those groups, I think they're using also MCP, agentic AI. Maybe there is a connection there. We're connecting your databases, your tools to the ones in the US.
And they use a dedicated registry, or do you know that?
Yeah, I think they're just starting using MCPs. But there's I think the question of who pays for those calculations, like when you make this again prompts. I mean, you can handle some like a small number of users, right? But after that, there is some cost associated with all these questions. Is that correct?
Yeah, that's a very good point. I actually want to say something about cost, and forgot to say thank you for reminding me. Go back to the slide. Where is it gone? Damn, I shouldn't have unshared here, I think. Share. Cost. Very good point. So it's something that worries me, cost. And I'll tell you why. So let's go back to the chat interface. Where is it? There we go. So what did we do? We did just a couple of prompts, didn't we? And it had to do some reasoning over it. And what we added is this little token login thing that shows the token log, which shows you how many tokens it used to generate responses and what the cost for that would be currently on our AWS instance. So now we paid roughly $2 to do those two things. So, I uploaded a paper, it extracted the genes and checked them in Netminer. I uploaded a GWAS dataset, extracted SNPs, genes, and I was searching in Netminer. So roughly, and we haven't really optimized it a lot. We're playing with caching and other things to bring it a bit down. So you can see we saved $1.33 because we used some caching, but it's certainly not something that we could just make public and free for anyone to hammer, and we would need to cover the cost. So I think we're building like a business model around it, which for free users, it will allow them to do 10 searches a day or 5 searches, and we would just absorb the cost. And then for premium subscribers, they would just need to, they pay like, £20, $50 a month, and they get 100 of searches. If they reach that limit, they would need to upgrade to the next plan to get more credits. So things it's hard, but it needs to be just thought about very carefully how to create a business model around this, because clearly there's a massive cost.
And for the connector, if we can also connect to like open Olama instance, if we have the infrastructure to run it.
Yeah, I think MCPI uses OpenAI, right? I think connection is there. Yeah, I think MCP is using OpenAI's tool, if I remember. And you don't pay for connection, but you pay for those.
Yeah,
that's true. But yeah, one is correct. I'm just. And Llama may have its own agentic AI tools. Yeah.
Yeah, sure. So definitely, if we are able to host something locally, and the open source models host locally and connect to them, then that would be probably a lot cheaper. But it still needs a server, it needs GPUs, it needs someone to maintain those things, keep them up to date, update operating system. So maintaining infrastructure has also cost, right? So it's just need to probably do a bit of a comparison, what is better. And then the open source models, are they as good as, in this case, we're using Claude 4.7 Sonnet. So it's a very powerful model. I have tried a few open source models that were good, not as good, but good, maybe good enough for some of these tasks. But here it's a generic interface. You could ask it to prepare presentations for you. You can ask it to, you can give it a Python script and it can do some custom analysis. You can run code as well. So not all of this you can do in a Llama model.
Of course. Yeah, but I think we have to think if we develop something like connectors to have the possibility to connect to different models, and then we can choose the financial one to host the thing or to pay. But yes.
I think at one point, I think for these models, for us, for example, WheatIS ranges, right? It will be like a Jabra, server side versus client side. Maybe the clients can pay for these tokens. So for WheatIS, that I think that should be the model if there's a very good.
Yes, I think we have to think on that. Yes, of course. But for the hackathon, for the hackathon, if we build the connector, after that we can put something on top or give the possibility to the community to put something. It could be us, but it should be another actor in China, so it will be open to the community if they want to pay with their own business model. But after that, we have to think also, and before that also to think what we want to use this connector for the WheatIS initiative.
Yeah, essentially like one user, 5 minutes, $1. Right.
Okay, so you have another set of slides. Let's go.
I can share the slides separately, Michael, and you can maybe distribute around. I just go to, so this was just giving motivation what agentic is and why is it nice to build an agentic system. Differences between the cost difference, we just talked about other differences between manual search and agentic search. The types of questions we could be aiming to answer. Some of them are listed here as part of a hackathon. I think it's good to not just focus technically, but what is the overall bigger goal we want to address? So what are the questions breeders are interested in or plant scientists are interested in, and some are listed here. And then define, as part of the hackathon, we can define, if the question is, what are the top wheat genes for salt tolerance linked to root development in wheat? Working together with developers and plant scientists, what would be the steps they would undertake to answer a question like this? And then we have to translate this into skill set an AI can understand. So let's just go to this slide. So hackathon format. Are we thinking about 3 days, did you say, Michael, for the hackathon?
Yes.
Yeah.
But
so
we have to discuss, but it could be it's a suggestion to have time to do something relevant.
Right. Yeah, I think 3 days is probably the minimum really to do something useful. So we can spend 1 or 2 days once we have defined the teams to define skills, schemas, build and test, and then day 3 to stitch things together, build workflows, try to answer some real wheat questions, and then give short 5-minute pitches or demos. And yeah, I heard the discussion earlier about incentives for people to join, maybe have an idea of a joint publication or a blog article, something that is open and public would be nice outcome of this, or put everything open source on a specific Git repository. That would be nice as well. And then, so there was 2 ideas that I had in mind, but I think it's important to collect those ideas and then see what people are really interested to work on. And then once we are ready to run the hackathon, we work on a specific task really targeted. So this was the agentic idea. building MCP servers with skills for these different databases that are listed here. The other one could be more on, I think, what Monica presented on extracting information from either abstract or full text, compare it a bit, show the added value of extracting information from full text rather than abstract, which models to use, and so on. It has lots of challenges as well. Could be a task. Other ideas? Yeah, that's it, I think. I'll share the slides and then you can discuss next few days you have all together. And
yeah, no, so we'll have to discuss with the members who are not attending the meeting. But yes, can I have some remarks from Monica or Sylvie, or do you have some comments? Paolo, do you have some comments on that? If plus the MCP and agentic parts that can interest you, or more the kind of more applied literature stuff, what do you think it will be interesting for you, for example, to do 3 days hackathon?
The agentic AI. I think the agentic AI is like the natural next step. I think, for example, we are already doing, I think a lot of us are doing stuff with LLMs, like in different sectors, but kind of all kind of sibling projects. So I think like the agentic AI would be an interesting thing to explore, like all together. And I don't have experience with it, but it would be fun to learn.
Yeah.
But also, LLMs still have lots of kinks to fix, and bugs and stuff. So also working that together.
Paolo, do you have something that should interest you in the ID presented by Kiwan?
I just agree with you all. I mean, the agentic, as Monica said, seems to be hot topic nowadays, right? So it makes sense to organize an hackathon on this.
Okay, so other comments? No, Mohammed, do you have any comments on that? You try to unmute.
Yeah. Yes. No, thank you very much. No specific comments from my side. Just one question, very short question. So suppose we upload a paper and find some genes related to some trade. Is it possible to connect that gene to other papers which have findings which are kind of validated? So at the end we know, okay, our findings are already validated somewhere.
Yes, totally. Yeah, you can search other papers with that gene list. Is that the question? So we extract genes and then we go and check a different knowledge base. What information does it have about those genes? And yeah.
Awesome. Thanks.
Okay, so thank you, Kiwan, for the presentation. We'll have to break up and to go to lunch. We can lunch until 1:30. So
okay.
we'll have to lunch. And thank you, Kiwan, for the presentation. I think that the MCP agentic stuff some big interest. So we'll move forward on this with the rest, all the expert working group, and to move forward. So thank you very much for all the attendants in the main room and all the attendants in online on a Sunday morning. So thank you very much. And I will circulate some summary on the video and so on. Thank you. Bye.
Have a great conference. See you guys.
Thank you. Bye.